You know, the biology we learn in school, it's often a super simplified story. Plants make food, viruses are just germs, and theories are, well, they're just guesses. But the reality is so much more complex, and honestly, it's way more fascinating. Today we're going to peel back those simple labels to see how science is constantly digging deeper, revealing these incredible truths that are hiding in plain sight.
Okay, let's start with a classic biological puzzle, one you might have wondered about yourself. Are viruses actually alive? I mean, on one hand, they replicate, they evolve. That sounds pretty lifelike, right? But on the other hand, they seem to break some of the most basic rules of life.
And the answer, it turns out, is it's complicated. Viruses are like these little biological ghosts. They can't reproduce by themselves. They're basically pirates that have to hijack a host cell's machinery just to make copies. They don't have a metabolism of their own, so it makes you wonder, are they alive, or are they just these incredibly intricate biochemical machines? This gray area, this ambiguity, is exactly what makes biology so cool.
Now, this kind of complexity isn't unique to viruses. And to really get it, we first need to challenge some of our most basic ideas about how science itself even works, starting with the very words we use. Here's a big one. How many times have you heard someone dismiss something important by saying, "Oh, that's just a theory"? It's a super common phrase, right? But it's built on this massive misunderstanding of what the word theory actually means to a scientist.
So in everyday talk, a theory is just a hunch. You know, like your theory about who took the last cookie from the jar. But in science, it's the total opposite. A scientific theory is this incredibly robust, well-supported explanation for why something happens. And it's backed by a mountain of evidence and rigorous testing. It's basically the gold standard of scientific understanding.
Okay, so if a theory isn't just a guess, does that mean it's set in stone, an unchanging fact? Not at all. Science isn't about finding a collection of final answers. It's this dynamic, ongoing process where we're constantly refining our understanding of how the universe works.
A lot of us were taught this simple ladder in school. A hypothesis grows up to become a theory, and if it's really good, it graduates and becomes a law. Yeah, that's not how it works. They actually have different jobs. A law, like the law of gravity, just describes what happens. It tells you that an apple will fall from a tree. A theory, like the theory of general relativity, explains why the apple falls. One isn't a better version of the other. They're just different, equally powerful tools for understanding the world.
Now, to see this whole process of scientific refinement in action, let's zoom in on one of the most fundamental processes on Earth, photosynthesis. We're going to see how our understanding has evolved from that simple classroom concept to a complex and, honestly, breathtakingly elegant explanation.
We all remember the middle school summary, right? Plants use sunlight, water, and CO2 to make food. Simple enough. But have you ever really stopped to wonder what is actually going on inside that leaf? How does a plant physically catch a particle of light and turn its energy into something useful? The reality is a stunning feat of nanoengineering, a microscopic world of unbelievable complexity.
And this is where it all begins. This beautiful, almost chaotic-looking structure is the engine. It's called Photosystem 2, or PS2 for short. Think of it like a microscopic machine, a bustling factory floor packed with proteins and pigments, all working together to perform the very first, most critical step, capturing the energy from a single photon of light.
For decades, scientists have been trying to create a detailed blueprint for how this tiny factory works. And just like our understanding of a theory, our picture of this whole process has gotten so much more detailed over time, moving way beyond those simple starting ideas.
Early on, scientists had a couple of really elegant, simple models to explain things. The lake model imagined all the captured light energy flowing freely across the entire system, almost like a wave in a lake. Then there was the puddle model, which saw that energy as being trapped in small, isolated pools. Both were really useful ideas to get the ball rolling, but as we're about to see, the reality is a whole lot more interesting.
One of the key questions was about the physical layout of these molecular factories. As you can see here, sometimes they're all mixed together in what's called a mixed layout. Other times, they separate into these distinct zones, a segregated layout. So the big question for researchers was, does this physical arrangement actually change how efficiently the whole system works? So, to find the answer, scientists built this incredibly detailed computer model. They were able to track the journey of every single packet of light energy across a tiny 200 nanometer patch of the photosynthetic membrane. And what they found? Well, it was remarkable.
And right here, this is the aha moment. Take a look at this graph.
That black line, that's the computer simulation, and the red dots, that's real-world data from actual plants. The match is almost perfect. And what's really amazing is that they achieve this with no free parameters, which basically means they didn't just tweak the model to make it fit the data. The model was so accurate it predicted reality all on its own.
So what did this powerful new model actually show us? It revealed that the energy moves in two totally distinct ways. Sometimes it zips around freely. That's diffusive transport, and you can think of it like a ball just bouncing all over a pinball machine. But at other times, it slows to a crawl in what's called subdiffusive transport, which is a lot more like trying to walk through a super sticky mud pit.
So what decides if the energy is playing pinball or wading through mud? It all comes down to the reaction center. That's the spot where energy actually gets converted. Look at the graph. When a reaction center is open and waiting for energy, it moves freely. See those top lines where the alpha value is close to 1? But once it captures a packet of energy and gets closed for processing, it becomes like an energy trap, and movement around it slows way down. That's those lower, flatter lines where alpha is less than 1. This incredible discovery allowed them to calculate a really critical number, the excitation diffusion length. The easiest way to think about this is as the effective range of a packet of light energy. It's the absolute maximum distance that energy can travel to find an open reaction center before it just fizzles out and is lost forever. And that number is just 25 nanometers. Now, to put that into perspective, a single human hair is about 80,000 nanometers thick. We are talking about an infinitesimally small distance, which means for this natural engine to be efficient at all, everything has to be packed together with breathtaking precision. And now we can see exactly why that membrane layout we saw earlier is so critical. In the mixed layout on the left, you see this sea of red. That tells us that most of the light harvesting antennas are well within that crucial 25 nanometer range. But now look at the segregated layout on the right. Those big blue patches show us collectors that are just too far away. They're basically stranded, and they're probably wasting the precious energy they capture. The final numbers here tell a really clear and dramatic story. That tightly packed, mixed layout, it boasts an incredible 82% efficiency. But the segregated layout, its efficiency plummets all the way down to just 70%. In the brutal world of survival, a 12% drop in your ability to make energy, well, that can be the difference between thriving and starving. So let's zoom back out for a second. We went from the simple idea of plants make food all the way down to the subdiffusive dance of energy across a 25 nanometer stage. This whole journey, from a simple fact to a complex reality, is the very heart of what scientific progress is all about. And this really brings us full circle. By challenging and dissecting that simple story that photosynthesis just happens, we're forced to think more critically. We replace an old, simple model with a new, much more accurate one, and by doing that, we gain this richer, more powerful understanding of how nature truly works at its most fundamental level. You know, understanding this hidden complexity isn't just an academic exercise. By building a better, more accurate model of photosynthesis, we can potentially learn to engineer more efficient crops or even design revolutionary new types of solar technology. And it really leaves you wondering, what other simple fact that we take for granted today holds the key to the next great breakthrough?